The California condors are critically endangered birds that face many threats, such as lead poisoning, habitat loss, and human disturbance. These majestic vultures are the largest flying birds in North America, with a wingspan of more than 9 feet. They can soar and glide for hours without flapping their wings, searching for dead animals to feed on. They are also long-lived, reaching up to 60 years of age. However, these remarkable birds have been pushed to the brink of extinction by human activities. In the past, they were hunted, poisoned, captured, and disturbed by settlers who reduced their food supply of large wild animals. By the late 1900s, only a few dozen condors remained in the wild in Southern California. They were also affected by contamination from lead fragments in carcasses, poison bait, and environmental pollutants. The pesticide DDT may have weakened their eggshells and prevented them from hatching. In 1987, the last 22 wild condors were captured and brought into a captive breeding program to save the species from extinction. Thanks to the efforts of conservationists, the population has slowly increased and some condors have been released back into the wild in California, Arizona, Utah, and Mexico. However, they still face many challenges in their natural habitat. Lead poisoning is a major problem that can cause illness and death in condors. They also suffer from habitat loss due to development and human disturbance. They are vulnerable to collisions with wires and structures, as well as predation by ravens and other animals. The California condor is a symbol of nature's beauty and resilience. It is also a reminder of our responsibility to protect and restore the environment that we share with these magnificent birds. By reducing lead use, preserving habitat, and supporting conservation efforts, we can help the California condor recover and thrive in the wild. A new vaccine developed by researchers at the University of California, Davis, could protect the condors from a deadly disease called West Nile virus, which has killed several birds in the wild and in captivity. West Nile virus is a mosquito-borne infection that can cause fatal inflammation of the brain and spinal cord in birds and other animals. The virus was first detected in North America in 1999 and has since spread across the continent, posing a serious threat to many bird species, especially raptors like eagles and condors. The California condor is one of the most endangered birds in the world, with fewer than 350 individuals left in the wild. The species was nearly wiped out by habitat loss, poaching, and poisoning from lead ammunition used by hunters. In the 1980s, the last 22 condors were captured and brought into a captive breeding program that has successfully increased their numbers and reintroduced them to parts of California, Arizona, Utah, and Mexico. However, the condors still face many challenges in their recovery, including avian flu, which has killed at least 22 condors in Arizona since 2022. To prevent further losses from West Nile virus, the researchers at UC Davis have developed a vaccine that is safe and effective for condors and other birds. The vaccine is based on a modified version of the virus that cannot replicate or cause disease, but stimulates the immune system to produce antibodies against the virus. The vaccine has been tested on chickens and pigeons, and has shown promising results in protecting them from infection. The researchers hope to obtain approval from the U.S. Fish and Wildlife Service to start vaccinating condors in the wild and in captivity as soon as possible. The vaccine could be a game changer for the conservation of the California condor, which is an iconic symbol of the American West and a vital part of the ecosystem. The condors are scavengers that feed on carcasses of large animals, helping to clean up the environment and prevent the spread of diseases. They are also culturally important for many Native American tribes, who consider them sacred and use their feathers for ceremonies. By protecting the condors from West Nile virus, the vaccine could help ensure their survival and recovery for generations to come. The vaccine is based on a modified version of the virus that does not cause disease in birds, but stimulates their immune system to produce antibodies. It has been tested on captive condors and shown to be safe and effective. The vaccine contains a killed, inactivated version of the virus that has been conditionally licensed since 2016. The U.S. Department of Agriculture granted emergency approval for the use of the vaccine after finding a California condor dead from highly pathogenic avian influenza, or bird flu, in March. Since then, at least 13 condors with bird flu have died near the Arizona-Utah border, alarming conservationists who have worked hard to save the critically endangered species from extinction. The vaccine will be administered to captive and free-flying condors in hopes of preventing additional deaths and protecting the population, which was estimated at 561 at the end of 2022. The vaccine will also help reduce the risk of transmission to other birds and potentially humans. The California condor is one of the world's largest flying birds with a wingspan of up to 9 feet and weighing more than 20 pounds. It is also one of the most vulnerable to threats such as lead poisoning, habitat loss and environmental pollutants. The vaccine is a vital tool in the ongoing efforts to conserve this magnificent animal and restore its natural role as a scavenger in the ecosystem.